Hey, Carp Geeks. I'm going to go through, I guess, an easy version of how carp are stimulated to feed by amino acids. Now, if you want more details, you can, of course, maybe you've seen it already, have a read of my longer article, Carp Fever Revisited, which is available. I'll put a link in the description. It's available at our website, so I'll also link that. Okay, so let's get started. And basically what I'm going to do, I've prepared a PowerPoint slide which I'll go through and I'll discuss the various points as we go. So let's start class, right? So first things first, we got to know really as anglers how fish actually detect and are stimulated by amino acids. And then if we know that, we can actually go on and make a product which will actually, provoke an involuntary feeding stimulation. That's been my life's work, which has been developing these products. Okay, so let's take a look. So first things first, fish, of course, have a nose, right? And there's a picture of the nares. The nares are, as you can see from the picture, and I'll kind of show with the mouse here, there's kind of an in and an outlet for each nostril, okay? And the water actually goes in, circulates through the olfactory apparatus, and then comes out the other side. So there's a continual wash of odors, if you wish, over the fish's olfactory receptors. And, we're familiar with classical smells, strawberry, whatever, right? But they also have a set of chemoreceptors in the olfactory bulb, which we'll talk about in a second, which detect amino acids specifically. Okay, so I'm going to show you kind of a gory picture, right? Hang on. So imagine we've kind of peeled back the layers of skin and we're doing a dissection, gory as it is, right? So the nares, which are the nostrils, are essentially here, and they're connected through an olfactory tract, or just basically a tube, to where all the olfactory recognition happens. This is the olfactory bulb. It's basically the organ the fish uses to smell, like we have in our nose, essentially. Okay, and then if we dive inside that olfactory bulb, and so this is kind of, I go here, take this kind of bulb here, inside we see these folds of what look like skin, and that's essentially what it is. That's called the olfactory rosette, the kind of the larger structure. And inside is what's called the olfactory epithelium, or skin. Now, if you've ever seen any of these documentaries about how food is digested, your stomach has lots of kind of like waves up and down, like, tines in a comb, which increases the surface area of the skin inside your stomach. Similarly, in your nose, that olfactory skin or epithelium has a very, very high surface area, and it's on that skin that the receptors actually sit. Okay, now this is an interesting picture. Okay, so imagine one of these folds of skin here. Okay, and we zoom in. This is kind of a side-on picture of that fold of skin. So up in the top here, this is pond water, right? So remember that water's coming in through the nares. The water from the pond is actually up here and brushing against this skin. Then we have this kind of actual skin itself, which is this kind of layer, and it's just a picture taken with an electron microscope, kind of neat. And as you can see, there's almost kind of these kind of stalks coming up through the skin. These are actually nerve cells, right? Okay, and if we kind of look at the top here, there's kind of a bunch of strands, it looks like, on the top here. And on these strands, which are called cilia, or if you like, fingers, think of the fingers of an anemone, right? It's on these fingers that the receptors actually sit. And if I look at that skin from the top down, that's the next picture, and we see these kind of little starfish shapes. That is the kind of the root, the center of each starfish is the root, if you like, of the nerve, where the nerve impulses are generated. And then we have these kind of little fingers, these little arms of the starfish come out. And that's kind of interesting. It's on those arms or cilia where all the action happens with amino acid reception. So finally, and that's about the limit of electron microscopes, we have to go in now and kind of draw pictures because it's just too small to see with a microscope. This cilia is like, think about a rubber glove. So put your hand in a rubber glove. Your fingers are inside the fish. The air is the pond water, if you wish. And there's a thin layer between the two. That thin layer, the rubber of that glove, is what we call the plasma membrane. So it's a very, very thin layer that separates the outside world from the inside world of the cell. Okay. Now, interestingly, this is how nature works. in the outside world, and we'll discuss it more in a moment, there are lots of sodium ions. Sodium periodic table Na. Na stands for natrium. It's the most abundant, literally, ion in nature, and it's everywhere. So there's lots of sodium in every water source. And this is why the oceans are so salty, because that salt eventually ends up in the ocean. So, natural water sources have sodium, and that sodium sits outside the cell, and we have these gates. So there's a gate here, a little passageway that goes from the inside world to the outside world. And as those sodium ions go through that gate, a difference in concentration across that barrier is really the origin of a nerve impulse or a food signal. And we'll talk more about that in a second. So that's the basic physiological review of how fish really are wired to detect amino acids. More detail in a second, but it's basically through allowing transfer of sodium ions from the surroundings. So if I go to the next page, we'll talk more about that process, right? So there's some familiar pictures. So there's our top view of the cilia, the starfish, if you want, and there's the side view of the stalks. And here's kind of a better kind of image, right? Okay, so these are the nerve cells. Okay, these are the cilia, right? And if we zoom in on one cilia, it's like a finger from a rubber glove. There are holes on the surface, but those holes are actually not holes, they're gates. Okay, so they're closed and they open when an amino acid sits on its receptor. Okay, and that's the key thing here, right? So if you want to understand how amino acids make a nerve impulse, this is where it's at, right? So if we look at these slides, we see how it happens, right? So these are the sodium ions in your outside world, right? Just sitting there, just in pond water, right? They're everywhere, right? Here is that gate, but it's closed. It's what's called a ligand-gated ion channel. Sounds like a hard description, but it's not. Ion channel, that's just the hole, right? Gate, it swings open and shut. Ligand, now that's just an amino acid. And ligand in chemistry is just this thing that binds to a surface, right? So when our signaling molecule that says up in the picture, which is our amino acid, sticks to the receptor, that gate opens and ions flow from the outside to the inside. That creates a voltage. You may know nerves are basically a voltage circuit, right? That gives that gives a voltage signal, and the circuit transmits that to the brain, and the brain, takes that as a food here signal, right? When the amino acid comes off, the gate closes. Now, the higher the concentration, the more of these gates are open, right? And when we reach that threshold concentration, this is essentially what impulse is designed to do. Impulse is designed to open, more details in a second, between 50 and 85% of these gates. When that happens, we get involuntary feeding response. So, sorry, class over. That's as much as detail I pretty much go into, okay, with the physiology of the fish. Now, how do we kind of use that information? So here we go. So this is literally the table taken from Carp Fever, and back to that Carp Fever Revisited article as a link. You can take a look at it in more detail, right? But what I've done, I've taken Kevin Maddox's results and I've kind of simplified them a little bit for us, right? So if we look at the best acting amino acid, it's valine and lysine. They, at the lowest concentration, they give the best response from fish in terms of making them feed. And then we have so that's the Premier League, right? Okay. And then we have, the Championship here, good reaction in League Two. And then we have, Vauxhall Premier or something, some minor league there. So really good reaction, and poor reaction. What that really means is how well the amino acid fits into that receptor site. If it fits really well, think about putting your hand in a glove you've owned for years, right? If it's a really good fit, it stays in there longer. It's what we call high residence time. Good fit, longer residence time, that channel stays open longer, more ions can flow from inside to outside, bigger signal. So that's why amino acids that fit the site well give a better response, okay? And to show you that, that's what we call a dosage response graph, okay? So it's kind of weird, but let's just talk through it. So what we've got is the percent of the receptors that are occupied by an amino acid on the left, okay? And then the concentration, essentially, of amino acids in the water. Now, that amino acid has to be in the water. It has to get to the fish's nose. That's a key thing, right? Okay, and the actual amount is much lower for the more potent ones. So the ones with a very good fit, if we look at the scale here, that's 0.1 ppm, and a ppm is a milligram per liter. So think of it like a tablet of drug, right? That's probably a 20 milligram tablet you got there, 0.2 of a gram, right, of active ingredient in there. So divide the amount of drug in your aspirin by 10, and that's how much is in a liter of water of amino acid to stimulate a response. Okay, and as you can see, for example, lysine is one of the most potent amino acids. It takes 0.1 ppm approximately to kind of see a signal here. And key thing here, it rises really rapidly. So it turns on quick, gets going, and then it quickly, what we call saturates. We'll talk about it in a moment. There's a very narrow band where it's optimal, right? But that's how the response works. When it turns on, it turns on. Okay. Turns on about, and it's orders of magnitude. So I need about 10 times as much as a good one to have the same response as lysine or valine, and then 10 times more, 100 times more than the really good one for the poor ones. So take-home message is here, literally any amino acid will work as a feeding stimulant. It's just the amount you got to be careful with, okay? Lysine and valine are the best choices if you want to make a commercial product, because you can make a very strong solution and anglers can use less for the same effect, okay? Interestingly, betaine, which everyone loves, a bit controversial, is actually quite poor. So you actually need quite a lot of betaine to have the same effect as literally 100 times less lysine or valine, which is kind of interesting. Now, this is where we get into the nuts and bolts. Okay, so when you look at literature for drug responses, think of it a bit like drinking coffee, right? You can have 1 cup of coffee in the morning and you're not quite there yet. You're not quite ready to start work. That, in terms of receptor site occupancy, is literally too few receptor sites are activated. You haven't got quite the kind of activation you need for a response. And I call that the too cold response. So if we look over here, if we have, and the black hexagons are receptor sites in this model and the green dots are amino acids stuck on those receptor sites. If I'm less than 50% occupied on my receptors, that's like 1 cup of coffee in the morning. That's just not going to do it, okay? And we call that too cold, right? So you can put amino acids in the swim. If you haven't got enough, the fish just aren't going to get turned on. It's too cold, all right? Now, the opposite of that is, of course, too hot, right? So if I put in too much amino acid, which is really easy to do given the concentrations that need to be there for this to happen. So if I go above about 0.1 ppm, 0.1 milligram per liter, it's too hot. It's like 20 cups of coffee. If you ever had 20 cups of coffee, like 6 espressos or something, you just don't feel right. You need to lie down. That's essentially what the fish do. If we ever overdose the fish, and that's the technical term, an overdose, you've heard it in medicine before. Now, if it's something not so nice, right, you have bad side effects. But for coffee and amino acids, the side effects are very mild. For amino acids, the fish literally just get dozy. They just kind of drift off, right? So not optimal for feeding, but it doesn't do the fish any harm, right? So above about 85% coverage, we're too hot. The fish are 10 cups of coffee in, right? So it turns out when you read the literature, and we've kind of confirmed this with our experiments, anywhere really, and it kind of turns on at 50%, anywhere between 50 and 85% coverage is what we call the Goldilocks zone. So if we get between 50 and 85% coverage of a single amino acid on the receptors of the fish, brilliant. It works great, right? And we call that the Goldilocks zone of optimal stimulation. So you can get 85 to 50% for literally any amino acid. You've just got to get the dose just right. And that's where the problem is. If you look at that graph, the narrow band that relates to the optimal stimulation, the narrow band of concentration is very, very narrow. That's a problem. You've got to get between 0.1 and 0.2 milligrams per liter of lysine, say, in your swim to have an optimal response. And that's almost impossible to do, okay, with single amino acids, because you have to have the dosing just right to get that Goldilocks zone. And not only is the Goldilocks zone very narrow, it's also at a very, very low concentration, Okay, so you can't be heavy-handed. So you're literally putting grains, you're counting grains in a liter. It's really, really, really fiddly and difficult. And that really explains why amino acids have been so finicky in the past. Sometimes you read anglers catch a bunch of fish, they get it in the Goldilocks zone just when they need to. Sometimes nothing happens, they don't catch any fish. All right, so it's always been a problem with amino acids kind of getting and staying in that Goldilocks zone of between 50 and 85% occupancy with the equivalent concentration range, which is really, really narrow. And that's always been the problem. And this has been something I've been wrestling with for 20 years, right? How on earth do we stay in the Goldilocks zone? And if you read Carp Fever Revisited, there's a long piece about, how it came to me in the middle of the night. It's kind of a eureka moment. But just to cut to the chase here, there's our ideal coverage, right? So that's from the previous page, right? Just right. That's our just right Goldilocks coverage. Remember, the black hexagons are the receptor sites and the green dots are amino acids, whichever one you choose, right? Now, here's the trick. What we do, we mix the amino acid, 50% say, if we want a 50% coverage, with 50% blocker. That's the green crosses on the picture. So if I mix a 50-50 blocker and amino, it doesn't matter how much now I throw at that set of 9 receptors. I could throw a million things at it. If half of them are crosses and half of them are circles, it's going to give me that every time, isn't it? So we've now moved from, this is the key discovery of our invention, we've moved from concentration dependence for Goldilocks zone to ratio. And ratios are really easy to control when we make the product, and that's what Impulse is, essentially. It's a just right mixture of blocker and amino acid to give you the optimal coverage, therefore optimal stimulation at literally any amount you happen to put in your bait. That's key. So there it is. So that's the phrase we use in our literature. Impulse simply locks in the just right fraction of amino acid coverage through the addition of a proprietary site-blocking molecular species. And that's our IP. That's our kind of special kind of invention. Okay. Now, proof in the pudding, of course, right? So over many, many years of development, we got this thing tested multiple times, both in-house, sent it out, and, we got it done with a legitimate independent study at Sparsholt College in the UK. There's a list of the thesis. You can go to the Sparsholt library and read it, or just go, and I'll put a link. You can go to my website, our website, aminotech.com, and actually read the actual thesis yourself if you wish to. And in that thesis, the researcher, Ollie, right, he found that Impulse gave more than twice as many bites as identical undosed baits, and it was proven with statistics and everything with multiple trials. So it's statistically proven 2-to-1 under ideal conditions. For you guys as anglers, that was a scientific study. We guarantee it will catch you more fish. So if you check the website out, we're not going to go all the way out there and say 2-to-1, but hey, we will guarantee more fish. Just give it a go and just follow the challenge. It gives you a description of how you would actually do a test, just like they did at Sparsholt, and you can prove it to yourself. We are so confident in this product, we know you'll catch more fish because it's proven science. Okay, let's move on. So a few take-home messages now. I've talked to anglers who've actually been using the product, and they've got some feedback, and it's useful. So I want to pass it on because there's a few kind of basic things that people just need to understand when using the product. Okay, first, Impulse 100% needs to get to the bottom. Okay, you need to get the Impulse to the bottom, assuming you're fishing with a bottom bait. It needs to get on the bottom, spread out. Okay, spread out. We can't then use it in a cloudy spot mix, okay? So you've got to either absorb it into something porous like ground bait, expander pellets, boilie crumb perhaps. That's why you have a liquid invention. Or if you have a harder bait like a boilie or particles like maize or something, we have a gel product and you can just put it on top. So the gel is quite viscous and sticky. It sticks on the bait, which takes it to the bottom. The last thing you want is to put the liquid on a boilie. Don't do that, okay? Because it will wash off on the way down, okay? Next one. You need to get that minimum concentration. You need to get all those receptors occupied with that ratio of blocker and amino acid. So, make sure you use the minimum concentration. Now, it's a trick. Why have I shown you a picture of a fjord? Well, when I was in Norway visiting in the fjord, the question on the boat is always the same question. How deep's the fjord? And the guide will always tell you the same answer. The fjord is about as deep as the mountains are high. Now, why is that pertinent here? Because you've seen these videos of people with other products spraying their boilies with this kind of like very kind of light, kind of goopy stuff, right? And it soaks in, and they come back a week later, more soaks in, and then, you know, they do the thing when they cut it open and it's soaked all the way through. How long did that take? A month. Hey, if it takes a month to soak in, how long do you think it takes to leak out? A month, right? So the mountains are as high as the fjord is deep. If it takes you forever to soak a bait, it takes forever to the aminos to come out. If that's true, you'll never get threshold concentration. So you need to establish that minimum. So you can't be soaking your baits for a long period of time with the liquid. It's got to be a surface coating with the gel if it's a hard bait, which comes right off on the bottom, or a very porous bait, which then just exudes it very quickly. Okay, and that is the big trick which anglers seem to forget. So you can't be putting the liquid in your base mixes, it just won't get out. And that was, I don't want to knock him because he's kind of a hero of mine, but Kevin Maddox actually discovered that amino acids stimulate fish to feed in the 80s. And then he put amino acids inside a boilie, boiled it and locked them inside. They never got out, and that's why it didn't work for him. Okay, so do not lock your aminos inside a boilie. Use an exterior coating on a boilie, or use a porous bait with an overspray of liquid. I personally like to use a PVA bag with pellets, which I spray with the impulse liquid before casting. You can also put like the gel on a boilie. You know, if you're using like just exclusively boilies, the ones you're just going to throw out, just quickly put some gel on there and then just get them out there. And finally, impulse, it is made of free amino acids, which are a food, right? It's basically a protein, or digested protein. So once it's out of the bottle, now in the bottle it's super concentrated, a bit like honey. Honey never goes off because it's so concentrated. Similarly with impulse, it will never go off in the bottle, but once you start using it and diluting it, essentially it becomes food for microorganisms. So you want to use it as quickly as possible, and we say, you know, within 8 hours. If you kind of start to smell sour, you know, when food starts to go off, think of it a bit like shelf life milk. Once you open that bottle that's been on the shelf in the supermarket for a month, you've got like, what, a day before your milk goes off, right? So think of it like that. Try and use impulse as soon as possible. Apply as soon as possible before fishing for best results. Okay, so there's a picture. Okay, last thing to finish up. Want to learn more? So, you know, there's some product information. We have the liquid available in four flavors: original, which is unflavored; fish, self-descriptive; sweet cream; and fruit. Okay, and we have it in liquid, paste, and gel. The gel's quite thick actually, so it does coat boilies, and it's great because you got an injector you can use to kind of, as the picture shows there, to kind of like poke it into PVA bags. It is PVA friendly, make sure that's well known. Okay, so the products are PVA friendly. Paste is awesome for a paste wrap. We got lots of feedback from barbel anglers who've been using paste wraps on the river. It, like, you know, it kind of like keeps the amino acids kind of in the area, even though there is a flow, which is good. Okay, so scan the QR code, biosourcebaits.com is the website. And as I mentioned, hey, we do back this up with a catch more fish guarantee. We call it the challenge. We do guarantee you'll catch more fish. Give the challenge a go. Feel free to email us directly at info@biosourcebaits.com if you've got any questions. All right, hope that makes sense. See you guys next time.